Hello everyone, we are back again with a very important topic of IGCSE, and this is one of the last chapters, that is genetic modification and a part of biotechnology. So we very well know that biotechnology is the process in which living organisms produce products for the benefit of human beings, and they are genetically modified. That means their genetic material is altered. That means their DNA is altered. So, how is this important for human beings and what important products are formed? One of the very important life saving products is insulin. Insulin is the hormone that lowers the sugar level. And you know that insulin is produced and is bought by people who lack in insulin. So, lack of insulin causes the disease called diabetes mellitus. So genetic material is modified. So in this video, we will see how bacteria is modified for producing insulin. So here are the steps. Let's see the steps. All right. So first of all, you have to have your gene of interest. What is the gene of interest over here? The gene of interest over here is the human DNA. So from the human DNA, you isolate the insulin producing gene. All right. So the insulin producing gene or DNA is isolated, right? So here you go. Now, thereafter, you have bacterium, which is the tool for genetic engineering. So inside the bacteria, you have its main DNA and extra chromosomal circular double-stranded DNA, which is called plasmid, right? So plasmid is actually the vector in genetic engineering. This is the vector. That means it carries DNA from one place to another. And it is easy to work with plasmid rather than the main DNA. So what do we do now? We have isolated the gene of interest, which was insulin. Then we opened up the bacterial cell wall. We isolated the plasmid. We cut open the plasmid with an enzyme called the restriction enzyme. And the same restriction enzyme was used to cut this insulin also. All right. Here they have not shown those cuts, but that's okay. Now see, restriction enzymes cut at specific sites. They cut at specific sites to generate sticky ends. Okay, they generate sticky ends. Sticky ends are the ends where the foreign DNA can stick and easily be sealed. That means they generate complementary sequences. So with the restriction enzymes, which are molecular scissors, you cut open the plasmid DNA. Okay, and now you are going to mix the gene of interest, which is the insulin producing gene, with the plasmid and the restriction enzymes. Okay, now in this step, you can see this blue part is inserted into the red plasmid. All right, now this generates a recombinant plasmid. A recombinant plasmid. Recombinant plasmid is the plasmid which has been genetically engineered and carries the foreign gene. Foreign gene is nothing but the gene of interest. All right? Okay. Now, you have genetically engineered the plasmid. Now you put it back into the bacteria. Okay, because you want to produce the product. The product will be produced when the genes are expressed. So you have genetically modified the plasmid. The plasmid is introduced into the bacteria. And this is called transformation. We transform bacteria. So the bacteria are put within a medium of these recombinant plasmids, and we see that they just take these plasmids. But it is not easy. There are always marker genes, etc., which are here and there. That is actually a part of A-level syllabus. For IGCSE, this much is enough. Okay, so the bacterial plasmid is inserted back into the bacteria, and this is grown on medium. So it is grown on medium, you can say cultured on medium. It is cultured on medium so that you generate different copies of these bacteria. Of course, there's screening process and all that is not a part of IGCSE right now. If you guys take ASA levels, we'll definitely put in videos for that. And then you propagate or allow these bacteria to multiply in big reactors called the fermenters, which has all necessary conditions called the media, the suitable temperature, the stirring, etc., etc. So this is, in short, the technique of genetic modification to genetically modify bacteria to produce insulin. So important keywords for IGCSE exam would be what is a vector, what is a plasmid, role of restriction enzymes, and of course, they also give you structure of the fermenter that we can do in the coming videos. Hope this is beneficial for you. Just, go through this chapter. You might get questions from this chapter. Thank you so much for watching. Stay tuned.